In this class, we are going to see the development of reproductive system. As we already know, the urinary and genital systems, they are closely associated in their development and function. Developmentally, they are complementary to each other as one system induces the development of the other one. So among the two systems, urinary system is formed first. So before going into the proper topic, that is the development of genital system or the reproductive system, I will just summarize the development of urinary system first. This diagram shows the section through the embryo after the folding process. So this area will be the head fold, this area will be the tail fold, and because of the folding, we can see the umbilical opening over this much area. This will be the midgut region, this is the foregut region, this is the hindgut region, and this roughly the gut tube will be connected to the externally lying secondary yolk sac through this duct portion over here, and this duct will be the vitreointestinal duct. Our area of concern for today's class in this section will be this structure, that is the allantoic diverticulum. This allantoic diverticulum is formed close to the hindgut region, and there is this extraembryonic mesoderm which is extending into the space between the gut tube and the allantoic diverticulum over here. This mesoderm will be extending further caudally as a septum, that is known as the urorectal septum. This urorectal septum will grow towards the cloacal membrane over here. If you remember correctly, cloacal membrane will be the area where there is no intervening intraembryonic mesoderm. So there will be only two layers. You have the endoderm and the ectoderm. Now the urorectal septum is growing towards the cloacal membrane, and that will be attached towards the cloacal membrane, thereby forming anteriorly lying urogenital sinus and posteriorly lying the primitive hindgut. So this area of the hindgut, posterior or caudal to the attachment of allantoic diverticulum, will be known as the cloaca. So cloaca is further subdivided into urogenital sinus and primitive hindgut by the development of urorectal septum. So after the completion of the development of urorectal septum, this is the arrangement. So the allantoic diverticulum will be becoming, it will be continuous with the region that is known as vesico-urethral canal, and here will be the area of urorectal septum, and this will be the area of the anterior part of or ventral part of the cloacal membrane. So this part of the cloaca, ventral part of the cloaca, further forms a structure that is known as the urogenital sinus. Now if you see the arrangement, we have the allantoic diverticulum and we have the vesico-urethral canal, then we have the urogenital sinus. If you look carefully, you can identify a duct is opening into the junction between this vesico-urethral canal and the urogenital sinus, and this duct will be the mesonephric duct. Now on further development, the caudalmost part of the primitive or vesico-urethral canal will be giving rise to the primitive urethra, initial parts of the urethra. The rest of the vesico-urethral canal will be forming the urinary bladder, and the urogenital sinus is further divided into a pelvic part over here and a phallic part over here. So this is the summary of the development. There will be endodermal cloaca, and that will be dividing into primitive rectum and primitive urogenital sinus because of the development of urorectal septum. And the primitive urogenital sinus is further subdivided into vesico-urethral canal and definitive urogenital sinus. This vesico-urethral canal will be giving rise to the urinary bladder in the cranial portion and primitive urethra in the caudal portion. This definitive urogenital sinus pelvic part in the cranial portion and phallic part in the caudal portion. What you're seeing here is a axial section. It's an axial section through the embryo after the process of gastrulation. We're not going into the details of this section. I just want you to focus on the centrally lying notochord in the midline, and on either side of the notochord we have the intraembryonic mesoderm. This is one side and this is the other side. This intraembryonic mesoderm will be further divided into paraxial mesoderm, lateral plate mesoderm, and the intermediate mesoderm. For the development of urogenital system, we are going to focus on this particular region, that is the intermediate mesoderm. Now this is a view from the lateral aspect after removing the anterior abdominal wall of the developing embryo. So if we look here, we can see the developing gut tube in this region. This is the vitellointestinal duct which is connecting the gut tube towards the secondary yolk sac. This is the allantoic diverticulum. This allantoic diverticulum, when traced caudally, we can see the vesicourethral canal, and we can see the urorectal septum is on developing towards the cloacal membrane. Once this septum, this septum develops into the cloacal membrane, once the formation becomes complete, the ventral portion will be known as the primitive urogenital sinus, and this area will be known as will become the vesicourethral canal, and this area will become the definitive urogenital sinus. Now we can see the blue colored duct over here, which is opening into the posterior wall of the urogenital sinus at the junction of vesicourethral canal and the definitive urogenital sinus. I want you to focus on this particular structure, which is an elevation produced or present in the posterior abdominal wall. This elevation consists of a lateral bluish portion and a medial reddish portion, and this elevation will be known as the urogenital ridge, and this urogenital ridge is actually developing in the intermediate mesoderm. In this particular urogenital ridge, the lateral portion will be forming the mesonephros, and cranial to this we will have the pronephros, caudal to this we have the metanephros. The medial portion of this will be forming the genital ridge, which is giving rise to the gonads. The pronephros will disappear. The mesonephros, that will persist, some part will persist and majority of them will disappear. The metanephros present over here will be giving rise to the tubular system, tubules of the developing kidney. So this is another axial section through the embryo. Here we can see the sectioning of urogenital ridge very clearly. We can see the gut tube. This is the dorsal mesentery. This is the body wall. So naturally this is the area of urogenital ridge. In this urogenital ridge, we can see the medialmost elevation will be the genital ridge, which is going to give rise to the gonads, and the lateral elevation will be known as, together they will be known as the nephrogenic cord, and this nephrogenic cord contains mainly the mesonephros and a duct over here, that is the mesonephric duct. Lateral to the mesonephric duct, we can identify the paramesonephric duct also. What happens within the mesonephros? Some excretory tubules will get developed within the mesonephros. These are the tubules over here, and the medial end of these tubules will receive branches from the dorsal aorta. These are the branches from the dorsal aorta, which is going towards the medial end of excretory tubule in the mesonephros. Laterally, this excretory tubule will be opening into this particular mesonephric duct. This is the arrangement in the mesonephros. Later on, the pronephros will get disappeared. They will get segmented and they will get disappeared. Some part of the mesonephros will persist, and mesonephric duct will persist entirely in the case of male. Now I want you to focus on this particular area where the mesonephric duct is opening into the posterior aspect of the urogenital sinus, and in the next figure is a further development. As we said earlier, majority of the parts of the mesonephros also will get disappear, but some parts will persist. That will give rise to the some of the tubules in the male reproductive system that we will see later. Now this is the lower segment of that previous section. I want you to focus over this particular area. So this was the mesonephric duct present in relation with the mesonephros, and this mesonephric duct will be opening into the posterior aspect of the urogenital sinus over here. Just before it opens here, it gives off a bud, and that bud will be known as ureteric bud. This ureteric bud will be growing into the metanephric blastoma over here, and this metanephric blastoma will be giving rise to the excretory tubules of the kidney. This ureteric bud will be giving rise to the collecting system, the major and minor calyces, the pelvis of the kidney. So I want you to focus this particular area. This is another schematic section of the embryo to show the urogenital ridge and the subdivisions. So we can see the posterior abdominal wall. This is the dorsal mesentery. This is the section through the gut tube, and on the side of the gut tube we have the urogenital ridge. If you look carefully, the urogenital ridge contains a medialmost genital ridge which is going to become the gonads, and we have the mesonephros, mesonephric duct, and the paramesonephric duct over here. This is another schematic diagram showing the further development of these ducts. We have already mentioned all these things. You can see the pronephros, mesonephros, metanephros, and we have the mesonephric duct which is opening into the posterior wall of urogenital sinus. Before it enters into the urogenital sinus, it gives off the bud over here. That bud will be the ureteric bud. Later, the lower parts of the mesonephric duct will get absorbed into the urogenital sinus. So urogenital sinus, as we already know, that is developed from the endodermal cloaca. So these are the lower parts of the mesonephric duct. We can see the ureteric bud emerging from the mesonephric duct just before it enters into the urogenital sinus, posterior wall of the urogenital sinus. Now after absorption, this ureteric bud will open into the urinary bladder separately. So the part of the mesonephric duct caudal to the origin of the ureteric bud over this area, that is the area that is getting absorbed into the future urinary bladder, that is the vesicourethral canal. Now because of this absorption, as we already seen, all these are opening separately into the urinary bladder. But these openings of the ureteric buds, we can focus over here. This is the posterior view of the developing vesicourethral canal, the developing urinary bladder. So these yellow dots are the ureteric buds, and later what happens, these ureteric buds will move cranially and laterally due to the continuous absorption of the buds. So there will be a triangular area formed on the posterior aspect of the urinary bladder, and this triangular area will be bounded on the upper aspect by the two ureters and the lower part by the opening of the mesonephric duct over here. Next, we will see about the development of prostate. So this diagram we already know. This is the cranial part of primitive urogenital sinus. This will forms the vesicourethral canal. This much parts. The lower part of the or caudal part of the vesicourethral canal will be forming the primitive urethra. This is the pelvic part of definitive urogenital sinus. This is the phallic part of definitive urogenital sinus. Now the prostate is developing from a large number of bud-like projections that arise from the epithelium of prostatic urethra. Prostatic urethra means the urethra that is going to develop from this particular part, the caudal end of the vesicourethral canal. And some part of the prostatic urethra arises from the cranial part of the pelvic part of definitive urogenital sinus also. These buds, they will form the secretory epithelium of the gland. The buds that are arising from the mesodermal part of the prostatic urethra, they form the inner glandular zone of the prostate. So as we already studied in the gross anatomy, in the prostate there will be an inner glandular zone and an outer glandular zone. This inner glandular zone will be developed from the mesodermal part of the prostatic urethra, and the outer glandular zone is actually developing from the prostatic urethra, that is developing from the endoderm, that is the source of outer glandular zone. This outer zone differentiates earlier than the inner zone, and in later life, as we already discussed in the gross anatomy, the outer zone is frequently the site of carcinomatous change, while the inner zone is affected in senile hypertrophy of the organ. The muscle and connective tissue of the gland, they are derived from surrounding mesenchyme, that is also forming the capsule of the gland. So we should be able to tell the embryological origin of different parts of the prostate gland and specifying their developmental source of the glandular portion also, that also developed from different areas. The inner one developed from mesoderm, outer one developed from the endodermal origin. And after puberty, they will undergo considerable development, and in old age sometimes it will undergo atrophy, but in some men it will undergo hypertrophy. It is rarely absent in the male. If we look at the female homologs of prostate, the counterparts of prostate in female, so there will be endodermal buds similar to those that form the prostate in male are also seen in the female. These buds, they will arise from the caudal part of vesicourethral canal and they give rise to the urethral glands. Whereas the buds arising from the urogenital sinus, they will form the paraurethral glands of Skinny. They are the female counterparts of prostate gland. We can move on to see the fate of the next duct, that is the paramesonephric duct in the female. So this is a section showing the urogenital ridge. As you have seen earlier, the surface of the urogenital ridge will be bounded or covered by the coelomic epithelium. So the coelomic epithelium of the urogenital ridge on the lateral aspect, they will invaginate into the urogenital ridge, and they lie lateral to the mesonephric duct in the cranial most part. You can see the process of invagination, and on further development that will form the paramesonephric duct. We will focus the paramesonephric duct in this particular diagram. This blue structure will be the paramesonephric duct. So we can trace them from cranial to caudal direction. In the cranial most part, this paramesonephric duct will be lying lateral to the mesonephric duct. And later what happens, when they are traced caudally, they cross to the medial side of the mesonephric duct. You can see the crossing over here. This structure will be the mesonephric duct. So here you can easily see the crossing over. Then after crossing, the two ducts will be joining with each other. You can see the point of fusion between the two ducts, and they form a structure that is known as uterovaginal canal. Uterovaginal canal. This uterovaginal canal, the caudal end of this canal will be opening into the dorsal wall of the definitive urogenital sinus. Hopefully you can understand this is the mesonephric duct that is opening into the dorsal wall of urogenital sinus. So in between them, in the midline, this uterovaginal canal also opening into the dorsal wall of urogenital sinus. And in the female, this particular part will be giving rise to, I'm talking about the part that is developing from the urogenital sinus, that is giving rise to the vestibule. And we can clearly understand from this diagram that this arrangement is almost looking similar to the arrangement of the female organs. Those are the uterine tubes, the uterus, and part of the vagina. Here we have the female reproductive organs. We can see the uterus, we can see the uterine tube, the lateralmost end of the uterine tube, we can see the ovary and the lowermost part cervix, the upper part of vagina. All those regions we can see over this diagram. So we can easily assume the source of their origin. So if we describe the epithelium of the uterus, that is developing from the fused paramesonephric or Mullerian duct, that is forming the uterovaginal canal. The myometrium in the uterus will be derived from the surrounding mesoderm, and as the thickness of myometrium increases, the unfused horizontal parts of the two paramesonephric duct over here, they come to be partially embedded within its substance, and that will help to form the fundus of the uterus. So once they get embedded, one part will be lying above it, so that part will become the fundus of the uterus. Now the cervix can easily recognized as a separate region in the lowermost part. So we have to remember, in the fetus, it's very important, in the fetus the cervical part is larger than that of the body of uterus. Now we are focusing on the unfused part of the paramesonephric duct that will give rise to the uterine tubes. The original points of invagination of the ducts into the coelomic epithelium will remain as the abdominal opening of the tubes. This will be the abdominal opening of the tubes. Fimbriae, the finger-like projections, will be formed in this particular region. We can see some of the anomalies in this figure because of the defect in the development of the Mullerian duct or the paramesonephric duct. The first figure, that is showing bicornuate uterus. That means the uterus is having two horns. This is the uterine canal. We can see the two horns of the uterus over here. The second figure, you can clearly see the uterine cavities are partially or completely getting separated, and this is known as it is separated due to a septum in the middle, and it is known as septate uterus. And if the entire uterine cavity, the cervix, the vagina, everything is separated, and this is roughly like the duplication of the entire system, entire organs. This condition will be known as uterus didelphys, uterus didelphys, and you can clearly see the indentation in between the two separate parts. Now the next figure that you have to note will be this condition where you have entire uterine cavity is present as two horns, but towards the cervix it is coming as a single region, and vagina is also single, and there will be a slight indentation in the middle, and this condition will be known as the uterus bicornis or uterus bicolis. If you look at this particular region, there will be a slight indentation in the cavity of the uterus. You can see the indentation in the cavity, and this condition will be known as arcuate uterus, arcuate. Now in this particular figure, we can identify the uterus and vagina. They will be separated into two, but there is no septal indentation over here, and that will be known as the subseptate uterus. There will be anomalies for the uterine tubes also. The tubes may be absent, they may be partially or completely duplicated. There can be atresia of the tubes also can be present. Now we will be seeing the development of the vagina. So this figure shows the lower end of the uterovaginal canal, which is formed by the fusion of the two paramesonephric ducts. This is the dorsal wall of the urogenital sinus, and you can see the point of junction between this uterovaginal canal and the urogenital sinus. Now the endodermal cells of the urogenital sinus will get proliferated, and they form two swellings, and these swellings will be known as sinovaginal bulbs. they will fuse to form one mass as soon as it formed. And later what happens? This is the fused sinovaginal bulb, and close to that, from the caudal part of the fused paramesonephric ducts, that's the uterovaginal canal, some part also will be developed, some structure will be developed, and together this entire structure, this much entire structure will be known as the vaginal plate. So as you can see clearly, major part of the vaginal plate is formed by the sinovaginal bulb, which is endodermal in origin, and the cranial part of the vaginal plate will be formed from the uterovaginal canal, that is mesodermal in origin. So the hymen is located at this junction where the lower end of vaginal canal is getting communicated or fused with the urogenital sinus. As you can clearly see, this surface and this surface, both the surface of the hymen will be lined by the endoderm, and vaginal cavity will be formed by the presence of development of a lumen within this vaginal plate. So when we look at the anomalies of vagina, you can see the formation of the vaginal cavity over here by formation of lumen within the vaginal plate, and that will be opening into the urogenital sinus. And when you look at the anomalies of vagina, vagina can be duplicated. It is usually associated with the duplication of the uterus that we have seen just before. The lumen may be subdivided longitudinally or transversely by a septum. Vagina can be absent. Hymen can be imperforated. Vagina may have abnormal communications with the rectum, that is rectovaginal fistula, or some abnormal communication with the urinary bladder, that is known as the vesicovaginal fistula. Now what happens in male? What happens to this paramesonephric ducts in the male? They will remain rudimentary in the male. Greater part of each paramesonephric duct will get disappear eventually. But the cranial part of the duct, they will persist as a small rounded body that is attached to the testis, and that will be known as appendix of testis, that may occasionally give rise to cyst. Now generally we assume that the prostatic utricle will represent the uterovaginal canal, and it is therefore a homologue of the uterus in male. We will move on to see the development of external genitalia in male and female. So this diagram is already familiar to you. So this will be the endodermal cloaca region. We can see the urorectal septum over here. That urorectal septum is developing towards the exterior, and at this region we have this membrane. That membrane will be the cloacal membrane, which is having endoderm and ectoderm alone. There is no intervening mesoderm. As the urorectal septum over here, which is developing towards the cloacal membrane, it reaches till the cloacal membrane, thereby dividing this membrane into an anterior urogenital membrane and a posterior anal membrane. And because of this development of urorectal septum or urogenital septum, we have the anteriorly or ventrally lying urogenital sinus and posteriorly lying primitive hindgut. Now we are going to look at this cloacal membrane from the inferior aspect. From this aspect we are going to see. So this is that particular view. This is the initial appearance. So we have the cloacal membrane, which is shown as yellowish color over here, and as the urorectal septum reaches towards the cloacal membrane, there is a linear groove appearance in the cloacal membrane, and along with that appearance we have two swellings which is heaped upon on the either side of the this linear groove, and these swellings will be known as the primitive urethral folds. So you can see the linear groove over here. On either side we can see the two swellings. These two swellings within this yellow colored region will be the right and left primitive urethral folds. In addition to these, On either side of the primitive urethral folds, there is another swelling, another elevation, which is produced due to the proliferation of mesoderm, and this elevation on either side will be known as the genital swelling or genital tubercle, and later on they will be known as the labioscrotal swelling. So we have a centrally like groove where the urorectal septum is attached towards the cloacal membrane. On either side of the groove, we have the primitive urethral folds. On further laterally on either side, we have the labioscrotal swelling or genital swelling. Then on the cranialmost part, here we have a tubercle, another projection, another swelling that is lying in the midline, and that is known as the phallic tubercle. Phallic tubercle. So at this stage, we have primitive urethral folds, genital swelling, and the genital tubercle or the phallic tubercle. Posteriorly, as we already know, this is the anal membrane, and in between them we will have the perineal body. Now we will see the specific development into male and female. First we will see the development of external genitalia in the female. So this is the particular development. As we can clearly see, the genital tubercle over here will become cylindrical and they form it will form the clitoris. The genital swellings on either side, lateralmost swellings, they will enlarge and they will form the labia majora. This posterior ends of the labia majora will be fused and they will form the posterior commissure over here. The primitive urethral folds on either side of this is one primitive urethral fold, this is another primitive urethral fold. These folds will become the labia minora, and in between the labia minora or primitive urethral folds, the urogenital membrane will break down. Now the established continuity will be present between urogenital sinus and the exterior. This is how the female external genitalia is formed. Moving on to the development of male external genitalia. In the male, this genital tubercle will grow enormously to form the penis. And as this genital tubercle grows, towards the tip of the penis there will be a sulcus. That sulcus will be known as the coronary sulcus, and because of the presence of or formation of this coronary sulcus, the tip will be differentiated to form the glans penis. Later the prepuce will be covering the glans penis, and that will be formed by the reduplication of the ectoderm covering distal part of this genital tubercle. Now we can see the primitive urethral folds over here. On the either side of that, we can see the genital swellings also. I want you to note this particular structure, that is the primitive urethral groove, which was the line of joining urorectal septum towards the urogenital membrane. As the phallic tubercle grows enormously in the male, this primitive urethral groove also will be growing along with it. You can see the enlarged primitive urethral groove over here, and since this is the external surface, this surface will be covered by the ectoderm, so this groove also will be covered by the ectoderm, and this groove now we can call it as primitive urethral groove. So in the subsequent slides we are going to see the further development of male external genitalia. So I will just explain this figure alone. This will be the primitive urethral folds. This is the primitive urethral groove, and on either side of the primitive urethral folds, this will be the genital swellings. This is the prominence formed from the genital tubercle, that is the phallus, penis in the male. This is a lateral view after taking a somewhat sagittal section. So you can see all these structures are arranged at this particular surface on the ectodermal aspect, and urogenital sinus is coming towards the urogenital membrane over this area. So this is the urogenital membrane. Now we will be describing these diagrams one by one. So in the figure A, you can see the phallus, and it is formed due to the enlargement of genital tubercle. Caudal to the phallus, there is a longitudinal depression that is known as the primitive urethral groove that we have seen already, and on either side there will be primitive urethral folds. Lateral to these folds we can see the genital swellings. That's what we have mentioned just now. In the depth of the primitive urethral groove, there is the urogenital membrane which separates the groove from the urogenital sinus. That membrane only we are seeing over here. Now in figure B, what happens? The phallus, the penis, has enlarged. The primitive urethral groove is beginning to extend onto it. You can see the primitive urethral groove, and this groove is extending into the phallus. Then a solid mass of endodermal cells derived from the urogenital sinus extends into the phallus. You can see the solid mass of endodermal cells over here. You have to note this is developed from the urogenital sinus. This mass is known as the urethral plate. That is labeled over here. This is the urethral plate. Now in figure C, this particular figure, as you can see, the primitive urethral groove is now fully formed. The urethral plate is also enlarged and extends deeper into the phallus. You can see the extension of the urethral plate into the depths of the along the length of the phallus. Now in figure D, you can see the urogenital membrane has been broken down. Here it is disappeared. Now the endoderm of the urogenital sinus can be seen from outside since because of the breakage of this urogenital membrane. Now you have to look at this figure E. You have to focus on the ectoderm overlying the urethral plate, that is over this area. The ectoderm overlying this urethral plate will get disappeared, and as a result of that, endoderm of the plate is seen from the surface. So on the surface, what we are seeing here is actually the endoderm or the urethral plate. Now in the next figure, we can see the center of the urethral plate is now broken down and convert the plate into a groove. So here it was a cord-like arrangement. Now since the disappearance of the cells in the urethral plate, this is now becoming a groove, and this groove now onwards you can call it as the definitive urethral groove, and the folds forming the edges, they are the definitive urethral folds. In this figure, you can clearly see the definitive urethral folds. They will grow toward each other. You can see the urethral folds over here, and they will grow towards each other, and they will fuse to form a median raphe over here. And in this way, the definitive urethral groove, it is converted into a tube, which is the urethra, the penile urethra. And this process is starting caudally and progresses cranially, the direction of the fusion. And in this figure, you have to note this specific area, which is the area of glans penis. And in this glans penis, urethra is not forming in the process as we have mentioned now. So there will be an ectodermal growth into the glans penis. They will form a cord-like structure. They will later canalized to form the urethra in the glans penis. Now they will be fused together to complete the entire formation of the urethra. When we look at the anomalies of development of male external genitalia, one of the important finding is the phimosis. That means the opening of the prepuce will become too narrow to allow the retraction of the prepuce. Now this condition where the urethra opening at the ventral aspect of the pineal shaft will be known as the hypospadias. That is due to the defective fusion of the definitive urethral folds. Sometimes the urethra can open on the dorsal aspect of the penis also, and that is known as epispadias. That is due to the defect in the formation of the anterior abdominal wall and associated structures, and usually that will be associated with the ectopia vesicae or ectopic vesicae. Now we will be seeing the details of development of gonads. First, we will see some general points regarding the primordial germ cells, the source of these primordial germ cells. This is an axial section of the developing embryo where you can see the developing gut tube over here. This area will be the dorsal mesentery of the corresponding gut tube. Now, this region will be the urogenital ridge, this area. We can see the paramesonephric duct over here. We can see the mesonephric duct over this area. These are the excretory tubules in the mesonephros. And on the medial most portion of the urogenital ridge, we have the genital ridge or the future gonads. So the primordial germ cells from which the future sperm and ovum that should develop, these cells are believed to be segregated early in the life of embryo. They probably differentiate in the wall of the yolk sac and they migrate to the region of the developing gonads. So from the yolk sac, they will be reaching towards the gut tube and they will be migrating through the dorsal mesentery, and through the posterior abdominal wall they will be reaching to this particular region of the medial aspect of urogenital ridge. All spermatozoa and ova that are formed throughout the life of an individual, they are believed to arise from these primordial germ cells. So the migration of primordial germ cells into this particular urogenital ridge, medial aspect of urogenital ridge, that is the genital ridge, this migration of primordial germ cells into this genital ridge will be essential for the development of gonads. These primordial germ cells will be having an inducting effect on the gonad. Now we will be seeing the development of testis. Each testis develops from the coelomic epithelium that covers the medial side of the mesonephros of the corresponding side. So we can see this is the midline. We can see the gut tube. This is the dorsal mesentery. So this is the coelomic epithelium that is covering the urogenital ridge, and this is the medial aspect of the urogenital ridge. So the testis will be developing from this area. In the region where the testis is to develop, epithelium will become thickened. You can see the thickening over here, and this thickening is known as the genital ridge. The cells of the germinal epithelium proliferate. We can see the proliferation, and they form sex coats. They are solid structures. They are sex coats. They grow into the underlying mesenchyme located over here. They reach deep into the gonad and they are called medullary cords. We can see the medullary cords over here. These are the medullary cords. They will become canalized to form the seminiferous tubules. Simultaneously, the primordial germ cells, they migrate to the region of the developing testis. These are the red dots. They are the primordial germ cells, and they get incorporated in the seminiferous tubules. We will see further details in the next slide. As we have said already, the canalization of the medullary sex cords will be forming the seminiferous tubules. We can see the tubules over here. These are the seminiferous tubules. Simultaneously, the primordial germ cells migrate to the region of developing testis and they will get incorporated into this seminiferous tubules. That is what we have seen in the previous slide, and here also we can see the primordial germ cells associated with the sections of seminiferous tubules. Now, in between the seminiferous tubules, we will have the interstitial cells. These cells are derived from sex cords that are not canalized. These interstitial cells, some of them, they are also derived from the surrounding mesenchyme. Now, what happens later? The mesenchymal cells surrounding the developing testis over here, this area, they form a dense layer of fibrous tissue. This fibrous tissue layer will be known as the tunica albuginea. It is labeled over here. It completely separates the sex cords developing seminiferous tubules from the germinal epithelium outside. So this epithelium can make no further contribution to the development of testicular tissue. Now we are going to see the ductal system of testis. So in this figure, this is the genital ridge, this is the nephrogenic cord, or this is the area of mesonephros, this is the mesonephric duct. So we can clearly see that the testis that is developing within the genital ridge region, gonadal region, is lying in close proximity to the mesonephric tubules and the mesonephros. As we already know, most of the mesonephric tubules will get degenerated. Some of them which lie closer to the testis will persist, and along with the mesonephric duct, they will form the duct system of the testis. The ends of the seminiferous tubules anastomose with one another to form something known as rete testis. So we can see over here, this is the developing testis, this is the area of mesonephros, and this will be the mesonephric duct. So we have seen the development of seminiferous tubules within the testis, and towards the mediastinum testis over here, the seminiferous tubules anastomose with one another to form the rete testis. We can see the development of rete testis over this diagram. These rete testis, they will establish contact with the remaining part of the mesonephric duct with some of the mesonephric tubules. You can see the connection made from the rete testis to the mesonephric duct over this area. And these mesonephric tubules which are present between them will be forming the vasa efferentia. The cranial part of the mesonephric duct over this area, they becomes highly coiled on itself to form the epididymis. You can see the epididymis over here. While its distal part, that will become the ductus deferens. This is the ductus deferens. The seminal vesicle arises on either side as a diverticulum from the lower end of the mesonephric duct. We already know the mesonephric duct is going to open into the urogenital sinus. Just before it enters into the urogenital sinus, it gives off the ureteric bud. But later on, this opening will get separated and ureteric bud will be opening into the urogenital sinus on its own. After that, another diverticulum will arise from the mesonephric duct. That will be the diverticulum for seminal vesicle. So the mesonephric duct between the seminal vesicle diverticulum and its opening into the urogenital sinus, which is the prostatic urethra, it will be forming the ejaculatory duct. The next topic will be the descent of testis. The descent of testis is important because the testis originally develops in the lumbar region of posterior abdominal wall, and during fetal life they gradually descend towards the scrotum. Roughly they reach the iliac fossa during the 3rd month, and they lie at the site of the deep inguinal ring up to the beginning of 7th month of intrauterine life. They pass through the inguinal canal during the 7th month, and they will lie at the superficial ring during 8th month, and they are normally in the scrotum by the end of 9th month. The descent of the testis is caused or assisted by several factors. They will be the first one will be the differential growth of the body wall. Second one, the formation of inguinal bursa. In this diagram, we can clearly see the inguinal bursa. About the 6th month of intrauterine life, the various layers of the abdominal wall of each side, they show an outpouching towards the scrotum. This is that particular outpouching. This pouch progressively increases in size and eventually reaches the bottom of the scrotal sac. The descending testis enters this pouch to reach the scrotum. You have to note specifically that the pouch is formed even before the testis enters it. The cavity of the inguinal bursa becomes the inguinal canal, and the various layers of its wall form the covering of the testis and spermatic cord. The next factor will be gubernaculum. We can see the gubernaculum over here. This is the developing testis. Gubernaculum is a band of mesenchyme which extends from the lower pole of the testis towards the scrotum. But it is very important to understand that the gubernaculum does not contain any contractile tissue. So the shortening of gubernacle is not possible. In spite of this, the gubernaculum does play an important part in the descent of testis, because when the embryo increases in size, the gubernaculum does not undergo a corresponding increase in length. So there will be a relative shortening of the gubernaculum, and this will help the testis assume a progressively lower position. The next feature will be the processus vaginalis. This is the diverticulum of the peritoneal cavity. We have seen this process already. Then the descending testis invaginates the processus vaginalis from behind, and after the descent of testis is completed, the processus vaginalis loses all connection with the peritoneal cavity, and that will become a layer of the testis that is known as the tunica vaginalis. So this is the diagram. You can see the processus vaginalis over here. Testis will be invaginating from behind. Now we can clearly see the processus vaginalis. It loses its connection with the abdominal cavity, and that forms a covering around the testis, and that covering will be known as the tunica vaginalis. Then lastly, the descent of the testis is also influenced by hormones secreted by the pars anterior of the hypophysis cerebri. Abnormal persistence of the processus vaginalis, it can lead to the formation of hernia. You can see the different types of hernia here in this figure. We have the interstitial hernia, we have the funicular hernia, infantile hernia, and also another condition we have to note at this point will be the hydrocele. That is the collection of fluid within this processus vaginalis or tunica vaginalis. Based on the developmental aspects, there will be different types of hydrocele also. If you think about the anomalies of development of testis, we will have anomalies of the testis itself. That means testis may be absent on one or both sides. Testis may be duplicated. Or there can be anomalies related with the descent also, and this condition will be known as cryptorchidism. Descent of the testis may fail to occur, or it will be incomplete. In this condition, the organ may lie in the lumbar region, in the iliac fossa, in the inguinal canal, or in the upper part of scrotum. Next important applied aspect, developmental aspect, will be the abnormal positions of the testis, which condition is known as ectopic testis. Ectopic testis, depending on the position, the names will differ. Then there can be anomalies of the duct system of the testis also possible. Now we are going to see the development of ovary. The early stages in the development of the ovary are exactly the same as in the testis. That means the coelomic epithelium on the medial side of the mesonephros, as you can see in the figure, that becomes thickened, and that will be forming the genital ridge. You can see the genital ridge. Then cords of cells or sex cords or medullary cords proliferate, and they will proliferate from this germinal epithelium, and they will grow into the underlying mesoderm. The primordial germ cells, these red dots, they are formed in relation to the yolk sac, migrate to the region of developing ovary through the mesentery of the gut tube, and they will give rise to oocytes. Upon reaching this gonad or ovary, the sex course becomes broken into small masses, this area, and the cells of each mass surround one primordial germ cell or an oocyte to form a primordial follicle. In this figure, we can compare the seminiferous tubule with that of the primordial follicles. This is the male gonad, this is the female gonad. Then, according to some textbooks, the original sex cords undergo regression in the ovary and they will be replaced by a new set of cortical cords arising from the coelomic epithelium. The interstitial glands, they differentiate from mesenchyme of the gonad. Since no tunica albuginea is formed, the germinal epithelium may contribute to the ovary even in postnatal life. We have reached the final portion of the topic, that we are going to see the fate of mesonephric duct and tubules in male and female. First, we will see the male. The mesonephric duct in the male gives rise to lot of structures. We have seen this in the previous slides. We will summarize it over here. So this will be the mesonephric duct. This is the area of the mesonephric tubules, and this is the developing gonads. This is the urogenital sinus from which the urinary bladder and urethra are getting developed. So if you look here, the mesonephric duct gives rise to the ureteric bud. The first structure will be the ureteric bud, and this ureteric bud will be giving rise to the ureters, renal pelvis, major and minor calyces of kidney, and collecting tubules of kidney. All of them will be derived from this ureteric bud. Then some part of the mesonephric duct over here will get absorbed into the urinary bladder, that is forming the second group of structure, that is the trigone of urinary bladder. Then along with this absorption, this mesonephric duct forms the posterior wall of the prostatic urethra, a part of prostatic urethra, cranial to the openings of ejaculatory duct. We have already seen that. Now, in relation with the gonads, this is the gonad. In relation with the gonads, the mesonephric duct will be highly coiled on itself to form the epididymis. Then, as a continuation of it, the ductus deferens over here. Then, on later development, after the absorption of this mesonephric duct into the urogenital sinus, another diverticulum arises from the mesonephric duct just before it enters into the urogenital sinus. This diverticulum will give rise to the seminal vesicle. Then after the seminal vesicle, this very small portion over here will be the ejaculatory duct, and when you consider prostate, the mesodermal part of prostate is also developed from the part developed from mesonephric duct. Then we have to consider another term, that is the appendix of epididymis. So appendix of epididymis, it's a small rounded structure that is attached to the head of the epididymis, and this structure is actually representing the cranial end of the mesonephric duct, this particular cranial end of mesonephric duct. Occasionally it may give rise to a cyst, and it should not get confused with the appendix of testis, which is actually a remnant of paramesonephric duct. So these are the different remnants of or the derivatives of the mesonephric duct in the male. Now we will consider the derivatives of mesonephric tubules in the male. So we can see the different structures in this figure, and we have seen the appendix of epididymis. You can see that over here. You can see the ductus deferens. We can see the seminal vesicle. We can see the part of urinary bladder which is developed from the mesonephric absorption of mesonephric duct. All these structures we have already mentioned. Now what we are going to focus is the the varieties of mesonephric tubules, as you already know, most of the mesonephric tubule will get disappeared, but some of them will persist and they form the vasa efferentia. Other mesonephric duct, they will form some vestigial structures that are seen near the testis. Usually they are unimportant. They become clinically important only when they give rise to the formation of cyst. So some of the remnants will be, first group will be the superior aberrant ductules. Second group of remnants will be inferior aberrant ductules. Then comes the paradidymis. So superior aberrant ductules, not afferent, aberrant ductules, or is otherwise known as epigenital tubules. They lie cranial to the vasa efferentia. You can already understand this is the area of the vasa efferentia. So this ductules will be lying cranial to the vasa efferentia. They are connected to testis alone, not connected to the epididymis. The next group will be the inferior aberrant ductules, which is known as the uh, which is connected to the epididymis alone, not to the testis. They lie caudal to the vasa efferentia. Now, in between the testis and epididymis, we have the paradidymis. Paradidymis, and this is otherwise known as the paragenital tubules. And this paradidymis is not connected to either testis or epididymis. So we have three set of remnants: superior aberrant ductules or epigenital tubules. They are connected to the testis alone. Inferior aberrant ductules, they will be connected to epididymis alone, and paradidymis, which are present between the testis and epididymis, but is not connected to any of them. Moving on, now we are going to see the fate of mesonephric ducts and tubules in the female. So we will move on to see the structures developed in the female. So as in the male, these mesonephric ducts, they give rise to the ureteric bud from which the ureter, pelvis, major and minor calyces, the collecting tubules of the kidney, all of them will be derived from the ureteric bud itself. And also this absorption of the mesonephric duct will be giving rise to the trigone of the urinary bladder. When you consider female urethra also, some part of the posterior wall is developed from the absorption of mesonephric duct. Other than this, we are going to consider the unique remnants present in the female, which is coming from the mesonephric duct and tubules. You have to understand these duct and tubules, they do not establish any connection with the developing ovary, but they will give rise to some vestigial structures that will be seen in the broad ligament near the ovary. We will see them now. So first one will be the major ones will be the epiphron and paraphron. So we can see the epiphron over here and paraphron over this region. So what is epiphron? It is a longitudinal duct. You can see the longitudinal duct, and it is running parallel to the uterine tube. You can clearly see the uterine tube over here. This is the longitudinal duct which is running parallel to the uterine tube, and you can identify a number of tubules, transverse ductules. They open into the longitudinal duct also, and this area is actually corresponding to epididymis and vasa efferentia of the male. In some cases, this longitudinal duct will be unusually long. It runs alongside of the uterus and lower down becomes embedded in the wall of the cervix, but it never opens into the uterine lumen. That will get embedded in the wall, but it never opens. And that is the equivalent of the male ductus deferens, and in female it is also known as Gardner's duct. Gardner's duct. Now when we consider the paraphron, it is actually consisting of small blind tubules. They lying between the ovary and the uterus. You can see that this is the uterus, this is the ovary. They are lying between the ovary and the uterus within the broad ligament, and that is the equivalent of female equivalent of paradidymis. We have seen the paradidymis in the previous slide. and this is the female equivalent of it. So these are the derivatives of mesonephric duct and tubules in the female. Now we can summarize all these derivatives into tables, that is the male and female homologues derived from undifferentiated genital system. So the indifferent gonads in male, that will be giving rise to the testis, female that will be giving rise to the ovary. The primordial germ cells, male it is spermatozoa, female it is ova. When you consider the cortex of the gonad, there will be sex codes. These medullary sex codes will be forming the seminiferous tubules in the male and the associated structures. In female, they will get separated and they will be forming the ovarian follicles around the germ cells. When you consider the medulla, here the seminiferous tubules will be in the male, that will be arranged to recte testis, and female, that is recte ovary, but that is not that prominent. When you consider the mesonephric tubules, mesonephric excretory tubules, they will become the vasa efferentia and paradidymis, but in case of female, it is corresponding with the epiphron and parophron. When you consider mesonephric duct, so when you consider the male reproductive system, that will be giving off appendix of epididymis, epididymis itself, ductus deferens, ejaculatory ducts, seminal vesicle, and when you consider the urinary system, that is present common to both male and female, ureter, pelvis, calyces, and collecting tubules. If you consider the female counterpart, appendix of ovary and duct of epiphron are actually derivatives of mesonephric duct. Now comes the paramesonephric duct or Mullerian ducts. So the remnant in male will be the appendix of testis and prostatic utricle. In female, that is actually forming the uterine tube, uterus, and upper part of vagina. Sometimes that give rise to paratubal cyst also. Now the gubernaculum testis, gubernaculum, that in the male will give rise to gubernaculum testis. In the female, that will become the round ligament of ovary and round ligament of uterus. The urogenital sinus in male, that give rise to the urinary bladder, urethra, prostate gland, and bulbourethral glands in male. In female also, the urinary bladder and urethra are developed from the urogenital sinus. In addition to this, we have seen some parts of vagina, urethra and paraurethral glands, vestibular glands will be developed from this particular urogenital sinus. Now we can understand the Mullerian eminence with regards to seminal colliculus, hymen in the female. Now we can go through the other important topics. The phallus, the urogenital fold, the labioscrotal swellings, they will give rise to the corresponding external genitalia part of male and female. So by that we will be completing this topic. So these are the important topics from this development of urogenital system. You should be able to describe the formation of endodermal cloaca and the derivatives of this cloaca. Second topic should be the derivatives of mesonephric ducts and tubules in male and female. You should be able to elaborate them. The third possible question will be anomalies in the development of Mullerian duct or paramesonephric duct. Fourth topic should be the development of male and female gonads. So you have to go through the entire topic, but give special importance to these topics. Thank you.